So up till now you have seen different events that take place during the process of replication. Let us see how this replication is initiated. So the initial formation of our replication fork requires the separation of the two strands of DNA duplex to provide the single-stranded DNA. So the first step in the replication is the formation of replication fork. So why this replication fork is needed? It is needed to make single-stranded DNA. Next question arises, why this single-stranded DNA is needed? This single-stranded DNA is required for DNA helicase to bind, because DNA helicases, they don't bind to the double-stranded DNA. They bind to the single-stranded DNA. So they require single-stranded DNA. Secondly, this single-stranded DNA acts as a template for the synthesis of both the RNA primer and the new DNA strand. So for these requirements, single-stranded DNA is required, and which is achieved through the formation of replication fork. Although DNA separation, strand separation, which is also called DNA unwinding, this is most easily accomplished at the chromosome ends. So at the end of a chromosome, you can see the DNA is double-stranded, and this is the end of, if this is the end of chromosome, this opening up of a double strand is very easy. So if replication has to start from the chromosome ends, the opening up would not be any problem. But DNA synthesis generally initiates at internal regions. It doesn't start at the end of the chromosome. So if this is a double-stranded DNA contained in a chromosome, replication starts from the regions between these ends. And as the circular chromosomes lack the chromosome ends, circular chromosomes which are present in bacteria, you know, there is no end in these circular chromosomes. So it makes internal DNA unwinding essential for the replication initiation in bacteria. The specific sites at which DNA unwinding and initiation of replication occur are called origins of replication, because there are many points where between the ends of the DNA molecules where replication can start. So these regions are called origins of replication. Depending on the organism, there may be as few as one or as many as thousands of origins per chromosome. There are DNA molecules are huge molecules, so thousands of points can be seen where the replication can start. So as in this figure you can see, this is a chromosome, a double-stranded DNA molecule. Here it is the point where the replication has been started. So this is origin of replication. Besides this point, in the same DNA molecule, in the same DNA strand, it is another point where replication is initiated. So here in a short piece you can see two sites or two origins of replications. So they move on these replication forks move towards the opposite directions, and finally the whole DNA molecule is replicated. It may start at 2 points, 3 points, hundreds of points, or even thousands of points where replication starts. Here that was the example of a linear chromosome, and here this is a circular chromosome. So in case of circular chromosome, there will be only one site, there will be only one origin of replication. As you can see, this is the point where replication will start. So this replication will grow towards both the directions, along the circular molecule of DNA, and finally at the end these single chromosome will be replicated into two chromosomes. So in this way the replication starts.